Before I start, I want to acknowledge the invitation. I'm very happy to be in Australia and being a bioinformatics community, as I used to be a bioinformatician before doing more deep learning stuff. Now, when I was a bioinformatician, most people were not familiar with GWAS or association, so I create a slide to talk a little bit about that, but I will go fast over it, but I just want to make sure that we use the same terminology. So one of the goals of human genetics is to understand the contribution of genes or DNA variation to a phenotype or disease of interest. And the way that we do it is that we collect large sample size of individual for a phenotype of interest. In this toy example, we collect an individual, we have high cholesterol level, low cholesterol level, and we look at their DNA, and we're trying to look if there's a relationship between the variation that we see with the phenotype of interest, which in this case is cholesterol. Now, for the first SNP, you don't see any pattern, but the second SNP, you clearly see every individual that have C allele have a high cholesterol level compared to the individual that have G allele. Now, you can actually formalize it. I like visualization, so you can visualize it. Here, the x-axis is the allele that you have for that SNP. The y-axis is the phenotypic value, and you're fitting this line. And if the slope of the line is significant, you will say that there's association between this SNP and the phenotype of interest. And the strength of this association is actually important. We call it effect size. It tells how strong there exists this association. And if you can perform this association test genome-wide, we call it genome-wide association study, or in short, GWAS, and it's extremely important in my view for personal health. It helps us to understand the disease. It helps us to create a genetic risk score very early on for someone's life compared to other risk scores that may not be applicable very early. And the most important for the purpose of this talk is that actually they're very important for drug discovery. Now, one of the challenges of performing large-scale GWASs is that we need large sample size. And for getting large sample size, we need high-quality phenotypes. And this is becoming more challenging because sequencing and collecting the genetic data is going to become more affordable. However, phenotyping and obtaining high-quality data is going to be the challenge. Now, we are currently in this setting that we are trading off accuracy with scalability. Like, for example, companies like 23andMe send you survey information, you fill it up, you send it back. So it's very scalable, but it's not necessarily accurate because you don't have great knowledge about some of the phenotype or some of the diseases that you have. On the other hand, if you go to the doctor and ask them, a clinician, to score a biobank, that would take a long time. You can't scale it to a lot of phenotype, but they're very accurate in some sense. So the goal of ML-based phenotyping is get to this point that you are scalable and you can have a highly accurate phenotype. Just to formalize it, the goal is that you use machine learning to approximate the phenotype of interest in a cheap and rapid way as possible. And obviously, we want it to be scalable. Now, how does this work? I will give it in a very high-level idea. The process is a two-step. The first step is that you're trying to train a model to predict the disease or the phenotype of interest using clinical data. So clinical data can be ECGs, MRI, fundus images, any data that clinician will look and try to predict the disease. That's called clinical data. And if you can be able to train this model, try to imitate what a clinician does, you can actually transfer this model to a data set that you have paired genetic data as well as clinical data. Data sets such as UK Biobank have this type of information a lot. And what you do is that you apply a trained model into the new data set, trying to create risk score for every individual you have in that data set. Now, if you have the risk score and you have the genetic data, we can run GWAS and do a lot of interesting analysis downstream that I would talk about later. As a proof of concept, we actually got interested in a disease called glaucoma. It's an eye disease. It causes that it affects your vision. And if you're not detected early, actually cause blindness. Actually, it's the third cause of blindness globally. And here I'm just showing an example of what will happen to your vision when you have glaucoma. Now, one of the biomarkers that actually doctors or clinicians utilize to say someone has glaucoma or not is a concept called vertical cup to disc, or in short, VCDR, is the ratio between your vertical and cup. You can estimate this from fundus images. So if you have been to your eye doctor, usually they take an image of your retina, and based on that, they can actually detect some of these diseases. Like you have glaucoma, you have AMD, other stuff. One of the metrics that actually they compute is this vertical cup to disc, or VCDR. And the first question we were interested in is that can we actually train a model that uses fundus images trying to give the VCDR score for every individual? And if we can do that, then we can actually run ML-based VCDR. Basically, get this trained model, apply it to every individual that we have in UK Biobank, because UK Biobank have a lot of individuals that have genotype data as well as fundus images, and you can create a score, the VCDR score for every individual, and then run GWAS. Now, as a proof of concept, we always want to compare to a baseline, and we were kind of lucky that a couple of months before we published our work, there was a group that actually utilized 2 ophthalmologists to grade every fundus images in UK Biobank. It took them 300 hours to do that, but they were able to grade every fundus images in UK Biobank. So we were like, this is the best way to compare our GWAS with their GWAS. Here I'm just showing, and Craig et al. Is a previous work that used 2 ophthalmologists. And you can see that ML-based VCDR not only detected what they had detected, but actually detected additional novel loci. Now, detecting novel loci is really good. It's good for drug companies. However, it should be relevant. We can have like some confounding or some other factors. It can be like our ML-based VCDR is capturing height, and height has a lot of loci. So we are very happy, but actually they're not related to VCDR at all. So we create a polygenic risk score, and we compare the polygenic risk score under 2 different models, p-value thresholding or last signet, and we compare it to the previous work. And we observed that actually in the UK Biobank data set, our polygenic risk score performs better. So this is great. We are very happy. But again, there can be a bias. We can be biased to one population or one data set. To make sure that that's not the case, we apply it to another data set, which is EPIC Norfolk. Now, unfortunately, still is white or British, but still is out of distribution. So we are a little bit happy that actually it's transferred to another data set as well. Now, we are very excited about this work. We published it a couple of years ago, and we decided to extend this work. One of the main bottlenecks of this work was that we make the assumption that we had access to high-quality labels to train a machine learning model. We had expert to label and train the first part of the ML-based VCDR. But what will happen if we don't have access to high-quality grade labels? And this is actually something that would happen in practice a lot. Particularly, we got interested in a phenotype called COPD. And the question we were asking, can we apply ML-based phenotyping to COPD? So what is a COPD? COPD stands for chronic obstruction pulmonary disease. It's a lung disease. It's the 3rd cause of mortality globally. It has a very strong genetic and environment component. Smoking is actually the main cause and is one of the causalities that we know causes COPD, but there's a very strong genetic component. And the first question we ask is that, well, how do clinicians detect someone has COPD or not? And the way that clinicians detect is that you perform a lung maneuver. Basically, you blow to a device called spirometry, and this device will try to collect your lung volume over time. And the output will be some figure like this, just to be fair, like this. And based on this figure, the doctor will say you have COPD or not. Now, for me, it's very interesting. They look at this curve, and I was like, wow, this curve has a lot of information. How do they do that? And apparently, there are 2 information that is very useful for doctors, is what is the total amount of your lung volume in the last second of the experiment. They're called FVC. And the other metric they use is that what is the total lung volume on the first second, which is called FV1 here. And I was, wait a minute, they look at only 2 numbers. They look at the whole curve, but only 2 numbers that is important. And actually, they get the ratio of these 2 numbers. They put a fixed threshold of 0.7, and they say you have COPD or not. And it's magic. I was very impressed. And I was like, the first question we wanted to ask is that, well, does the whole curve has any additional information? So we said, let's train a model. Let's give the whole model to a deep learning network and see if it can predict someone has COPD or not, and see if what is the model is trying to learn. To answer, we actually use a model called ResNet. ResNet is actually a very popular model in vision, and particularly deep learning. I won't get into detail of the architecture. If you're interested, we can talk offline. But I think one of the novel things that this model had, it has a concept of skip layer. So traditional, when you have a neural net, the information has to go from the first network layer all the way to the last layer in order to last layer start training. However, by having skip layer, you can actually skip some of these layers and try to make sure that the downstream layers start the training before like the first few layers make their own training. And this is actually a very cute idea because it's simple, it makes the training really fast, and actually really improve the result. So we tried to use this model, although we use ResNet 18. 18 just says that then how many blocks are there. We usually envision this model is much bigger, but the data is simpler. Curve doesn't look that complicated, so we didn't have to use something super complicated. So we trained this model. We performed the same concept. We applied it in UK Biobank. Now UK Biobank has spirogram for all the individuals, which is great because like you have 500,000 individuals that you have genetic data and spiro, and you can create risk score for COPD. And we run GWAS. But before I show the GWAS result, the first thing we have to do is that, is this risk score is a good risk score or not? This ML model, does it doing a good job, or we think it's doing a good job, but then and GWAS will say something, but we are not sure. To make sure that actually our result makes sense, we actually compare to what clinicians use or other metrics that we can actually utilize from this curve. And I'm showing here the result of our ML-based model on different categories. On the COPD status, COPD death, and COPD hospitalization, in all of these categories, our model performed better. So it shows that it's a better risk score, but it doesn't mean that it's really the right risk score because it may be capturing or confounding to some other factors. So we want to really make sure that this is a real COPD score. And I feel like one of the ways to do that in my context as a genetic person is that to look at the genetic signal and see if this genetic signal tells me something about this risk score. So we compare our GWAS with the largest GWAS that was done on UK Biobank for COPD. So here I'm just showing the comparison of the significance score. So the y-axis is the minus log p-value of ML-based COPD, our model. The x-axis is the minus log p-value of the largest GWAS. And every dot represents one variant. And obviously we are clumping based on LD, where will be too many dots in this plot. It's going to be annoying. But you can see that most of these points are shifted towards the y-axis, indicating that the strength of association is stronger in ML-based COPD, which is great. It indicates that the power in ML-based COPD is higher. But again, this can be confounding. This can be, we are capturing height, and we are very happy that we have a strong GWAS. To make sure that this is actually a real COPD signal, we have to compare the effect size. And we did that. So here we compare our effect size with the largest GWAS effect size. And you can see there is a good concordance. I feel like there is like this R squared is actually indicating that our effect size is actually really similar to the effect size that was computed in the previous works. And we were trying to publish this work. One of the reviewers asked a really interesting question. He said, well, you guys are making a claim that you have an ML-based risk score. Why don't you run this risk score for only healthy individual in UK Biobank? You should be able to still give a risk score. You still get a spectrum. You still be able to run a GWAS. And what will happen to that GWAS? And I think it was a brilliant idea by the reviewers. I was very happy that they raised this question. And we run this experiment. And the experiment showed us that actually the effect size is very similar to when you run a GWAS on all the individuals in UK Biobank. And kind of it makes sense because if it gives a real distribution, the effect size shouldn't matter that much. And for me, this question made me to learn a little bit more about the work we were doing. It teaches us that actually in data sets that are actually biased to healthy, we still can run ML-based phenotyping. Honestly, before this question, I thought that always if a data set has too many healthy individuals, it would be very hard for a disease because we don't have that much disease. So this idea shouldn't work, but it seems that it works. And I was very happy about this question. Now, up to this point, I have talked about cases that you can run ML-based phenotyping when you have label, either high-quality label or some noisy labels. The next question we were interested in is that what will happen if you don't have label at all? So I'm going to talk about a method called REGAL, stand for Representation Learning for Genomic Discovery on Lower Embedding. So just reiterate, we want to run GWAS on a high-dimensional clinical data. These can be ECGs, this can be MRI. In this context, is spirogram. So spirogram has 1,000 points. The question is that how can we run GWAS of 1,000 points and we have no disease label? But spirogram captures traits that are related to lung. So any lung diseases should be captured in this data as well. So one simple solution is to run GWAS for every point. You have 1,000 points, you run 1,000 GWASs. Now, if people who have run GWASs are not that fast, it will take computation, it's slow. And the annoying part is that you're redoing the computation over and over. A lot of these points are actually highly correlated, so why should you run the same GWAS over and over? Now, I don't think so. The computation is the most problem here is that you're increasing your hypothesis correction, and you have to somehow correct it. Now, you can say, well, I'm smart. I know that I'm doing a lot of tests. These tests are correlated. I will figure out the right value, and then the way I'm going to combine them. This makes it really complicated. However, you don't have to design any new method. You just run the GWASs. So that's maybe the advantage of it. I think the other clever idea you can run that actually people have proposed is to run a PCA, do a lower embedding of the high dimensional data, and run GWAS on the lower embedding that you learn. And these lower embedding are the latent factors or latent embedding of the data, which can be important, and actually they're very important. People have shown that it gives you a lot of good results. Now, PCA has one big problem or assumption is that makes an assumption that there is a linear relationship between the latent factors and the input data. And we know in nature, biology is not necessarily linear. It has a lot of nonlinearity. And the question is that can we actually come up with a method that tries to model this nonlinearity that may exist between the latent factors and the input data? And still, we want to have this interesting property that PCA has. We want to make sure that these latent factors are somehow disentangled, because you don't want to capture the same signal over and over. So we want to have the nonlinearity, but we want to make sure that the embedding we are learning are somehow orthogonal for each other. So as I said, we propose a method called REGAL, and I won't talk about the acronym, but you have a high-dimensional clinical data. You perform an operation, which I will talk about it, is variation autoencoder. You get these latent factors, and from the latent factors, you try to reconstruct the input again. And your goal is to make sure that this reconstruction is very close to your input. So you have to have enough latent factors to reconstruct it again. Now, if you can do that, you can get these latent factors, run a GWAS, and start getting some understanding of what these latent factors tells you. But the most important part for me is that if you can do this, you can create polygenic score for every latent factors, and then you can actually try to learn a polygenic risk score for a disease using a very small sample size. Because you assume that there is a linear combination of these latent factors that if you combine them, you can predict a disease such as asthma if you're dealing with lung, or COPD. And you don't need that many individuals to train a polygenic risk score for asthma. You need a lot of high-dimensional data, but those are actually very easable in healthcare data. You can access them. So just a little bit of background about autoencoders. Autoencoders actually consist of two networks. The first part of the network is called the encoder, gets the input data, tries to summarize the data in a latent space. So the first part of the encoder is just learning the embedding. And the second part of the network, which is the decoder, gets the embedding and tries to reconstruct the output. And the output and input should be very close. That's what the loss that you used in this autoencoder is, that you don't want to minimize the difference between the input and output. Now, it's a very powerful model. It has a lot of interesting stuff, but one of the properties that it doesn't have is the latent that it learns doesn't have to be independent. It doesn't have to be orthogonal with each other. Actually, there's no assumption. But there is another model, which is probabilistic and stochastic, called variation autoencoders. And it's very similar to the autoencoder, but has this interesting property that instead of outputting the latent factors as part of the encoder, is actually output a mean and a variance for a distribution. Now, you can make an assumption what distribution you have, but if you have a distribution, it will predict what is the mean and the variance of distribution. And from this mean and variance, you can sample your latent factors. Now, the way you make an assumption, what is your distribution and how you sample, you can actually make these latent factors to be independent from each other. So if you look at the loss, it has the reconstruction loss, which is very similar to autoencoders, and it has the loss that tries to make sure that the latent factors actually Gaussian, follows a normal distribution based on this KL diversion loss. Now, I won't go into the detail, but that's the main idea behind. Now, we apply it to spirogram. Here, I'm just showing the architecture. I won't go to the detail, but the encoder consists of 3 convolution neural net, and it's connected to a multi-layer perceptron. That's the encoder part. And the decoder is actually very similar to the encoder. The only difference is that the order is reversed, because you want to reconstruct this back again. Now, this is, again, we call it this embedding called SPINC, stand for spirogram encoding. It does a pretty good job. I won't show the result, but if you're interested, you can look in the paper. But what we were ignoring is that all this knowledge that clinicians have, doctors and clinicians have been looking at this data for so long. So they have a very good intuition, what are the important features? And the question is that can we actually incorporate these features in our model? So we kind of change our model. We come up with the new name for the embedding called RSPINC. And the goal is to inject the information that doctor or clinician have to this network. So the encoder part is not changing. However, the decoder, in addition to our latent vectors that is created, we are injecting these features that doctor know. Particularly, like for example, FE1, FEC that were important for spirogram, they get injected to this model as well. And the model, what it does is try to learn additional latent factors on top of what doctors or clinicians know. So as an example, here I'm showing a result that I'm showing the first plot on the top actually is comparing the reconstruction of the true with what created. You can see actually the curves are very similar, which is doing a good job. The second curve on the top is showing the result of PCA with our model. You can see that when the latent factors are small, the model works really well, but after a point, they get very similar. And you can imagine if you use all the 1,000 point, both methods should have very like 0 error because you should be able to construct it. However, that's only true for training dataset. Now, I'm showing the R-spink result here, and that shows like when we manipulate the value of R-spink, what will happen to the spirogram curve. You can see it affects it. But what is important, what's the main message here is that R-spink do exist, means that there are latent factors on the high dimensional data that actually is not captured by the feature that doctor are using. And this kind of go back to my ML-based COPD, that when you utilize a full curve, you do better than clinician because there's some information in the curve that doctors or clinicians are not using. Now, I can't not show a Manhattan plot in a genetic talk. So here is the Manhattan plot of a genetic when we run GWAS on R-spink. And you can see that actually there are a lot of hits and there is a strong heritability. It's like 16% heritability. And we were very excited that this there for R-spink. That means that there is a genetic component for one of these latent factors that is not captured by doctors. And finally, we really want to make sure that actually we can create a polygenic risk score for asthma using a very few sample size, and we compare it to what doctors would do if they use the spirogram. And you can see actually this, the asthma PRS performs better and significantly better than the doctors. And if you use the doctor information plus the 2 spinks, you further do better. I think at this point, I would try to summarize the talk. Hopefully, I convinced you guys that ML-based phenotyping can work for certain phenotype if you have high quality clinical data. I show it under 2 different settings, on a supervised and unsupervised. In the case of supervised, I show if you have high quality data, such as VCDR, you can perform ML-based VCDR. However, if you don't have high quality labels, you have access to billing code, which maybe is very common in US, or self-reported, you still can run ML-based COPD. I talk about the method called Regal, that if you have no access to label data, you can still run GWASs, and you can get a very interesting result and trait understanding about the tissue that you're analyzing. Now, I didn't have time, but health data is multimodal. We don't get one modality, we get multiple modality, and we currently have a manuscript that actually tries to learn this multimodal in a one joint analysis. And we call it a multi-regal. If you're interested, I can talk more later offline. I would acknowledge the work. This is the work of many people. I was fortunate to come and present here, and I'm very grateful for their work, as well as our collaborator, which are amazing clinicians and help us to increase our understanding about different problem and different disease. And thanks for the organizer for giving the opportunity to present here. And I'm more than happy to talk about any of these projects or project that you can think that AI can help. So thank you for your time.
Thank you, Farhad. We already have questions. Okay.
Hi, thanks. That was a really great talk. I just had a quick question, and I don't know if Regal kind of can be applied to this, but for many disease phenotypes, it's a single diagnosis, but actually it's a very heterogeneous phenotype in terms of the underlying biology. So it's different underlying biology that eventually leads to, I guess, very similar symptoms that are presented, and they get the same diagnosis. And I was just wondering, have these approaches been applied to identify new subtypes of disease, and have you done that already?
No, we haven't done it, but we really want to do it for asthma, and the work started. And as you said, our first stab at it is to use Regal, but there are other high dimensional that we can use. But short, we haven't done anything, but we are very excited to do that. Yes.
Okay, thanks. And I'm really keen to talk later, so
Sure.
I'll come and find you.
Yeah.
Hi, Thank you so much for the talk. I have more of a general question, because you say you collaborate with clinicians, and I'm just wondering how do you approach the kind of lack of explainability of deep learning models, of making it they're very tricky to explain, and I'm assuming clinicians want to know why it predicted this and not that. How do you approach this?
So I think for each of these, we have to take a very different steps. Particularly, like for example, in the COPD, our collaborative clinicians were on board from the beginning, and they had a good understanding. So we did have to run some explanations and tell them that, hey, we think the model is focusing on this part, and for them, they would say, oh, this makes sense, or this doesn't make sense, right? But I think what they really got excited, because they were geneticists. I think that was one of the big plus in our case, that our clinician, not only they were like doctors, they actually were doing research in COPD. When we showed the Manhattan plot and it hit, they were like, okay, this makes sense. I expect to see these. And particularly when we show the effect size comparison with actually the GWAS that they run, they got really convinced that this is like really COPD. Now, if they get convinced to go into their hospital and ask them to apply it, it's something they want to do, but they're worried about some of the legal and other stuff. But it will take much longer time to do that.
Thank you.
Thank you so much, Farhad. That was quite interesting talk. My question is about the interpreting the functional impact of the genetic variation, how your deep learning model, they can help to interpret the functional impact of the genetic variation that are associated to genetic diseases. And the second question is regarding the co-founding and the dependency when you are dealing with genomics and phenotype in deep learning. Co-founding and dependency are the most important pitfalls. So how did you deal with this problem? Thank you.
So for the first question, to use deep learning to interpret variants, like our work hasn't done that, but there are a lot of works that does it, particularly like you can predict gene expression or parameterize, or like what's the effect of protein. So short answer, we are not doing that. I think that's the downstream analysis. We do run other folks' method if we want to understand the interpretation of some of this, but generally we talk with clinicians to figure out. The second question about confounding is a really hard question. You can never be sure that what you're doing is correct, in a sense that the model is learning something. However, for me, I got convinced when I look at the effect size, particularly at ML-based COPD, that the effect size replicate the known GWAS in some sense. So it's not capturing some confounding. The second thing I highly recommend is to look at your input data. That's why we always have this, like we look at 1,000 random inputs and see, for example, in the case of Regal, how did it reconstruct? And did it reconstruct okay? Or there was some issues? Or if you're dealing with images, definitely open the file, look at the images if you can. Sometimes you don't have the legal to open it, but if you can, open it, look at it. Because sometimes doctor writes something on the images and you want to make sure the model is not learning that. So you have to do a lot of stuff. Like I would highly recommend look at the input data and make sure that there is no confounding there. Now, about the confounding on GWAS, I'm less concerned. There are some concerns sometimes, but at least for some of these analyses, I'm less concerned that there's a confounding on the GWAS side. But the ML side, you have to do a lot of analysis to convince yourself, I feel like, to make sure that this is not learning something. Now, one good thing about running the genetic analysis for me, if there was a confounding that is not a genetic confounding, then you shouldn't see any genetic signal. That will completely should mess, like if it's learning